in this PCR, Columbia didn't get amplified along with a bunch of the other samples in the first reaction. This led us to have inconclusive data for that line. So here's the table that summarizes the results that we got from our genotyping. In all, we found 5 homozygous mutants, which we will grow to see the effect of the mutation on secondary cell wall biosynthesis. So here we can see the data that we compiled from our tomato root measurements. This is a graph showing the mean root length for each intergression line 5 days after germination. And each of these ILs are compared to M82, which is glycopersicum. And what we're trying to do is to find the ILs which have a root length that is statistically different from M82. And as you can see here, these two are the most statistically different. And we're going to use these ILs to find QTLs in the future to identify the regions of the Pinelli DNA that drive the phenotype that is seen in the M82. So here's a quick comparison. We can see this root, I believe, that is 4-1, 5 days after germination. Here is M82, 5 days after germination, and Pinelli 5 days after germination. You can clearly see that 4-1 is much longer than the other two. So we're going to take our next steps based on this research, and we're going to do this by first looking at the ILs as quantitative traits in more detail in tomato. We're going to find QTLs, the quantitative trait loci, to find out which loci regulate the phenotypes that we're measuring. And in Arabidopsis, now that we've found the mutants, we are going to grow them to see the effect that the mutations have on the phenotype and the genotype of the plant. This will also allow us to validate the gene network that members of our lab have created in the yeast 1 hybrid. So the applications of our research are also profound. Our Arabidopsis gene network will help further our tomato research, as there are many homologous genes between the two plants. Also, both of our research topics can help us find the best variety of plants for different situations. In Arabidopsis, our research will help us find the varieties of plants that have more secondary cell walls. This is beneficial as secondary cell walls help take up water through the xylem. Thus, it will help plants grow better. Also, secondary cell walls add more sugar to the plant cell walls, which is very beneficial for biofuel companies which are seeking to use plants as their source of alternative fuel. As for tomatoes, we can use our research to find the varieties of tomatoes that grow best in different situations, so we can expand the growth of tomatoes as tomatoes are commonly eaten around the world. The varieties of tomatoes will also help us find a good variety for use in agriculture. So I would like to thank the Brady Lab for all their help and support and the materials that they provided for the experiment. I'd also like to thank Dr. Brady and Dr. DeLucas for all their lessons and their support and for putting up with my zillion questions. I'd also like to thank my parents for all their support and YSP for giving me this opportunity to do research. I'd also like to thank Jess for being an awesome counselor. So now I'm open for questions.
When you're first talking about the Arabidopsis plants, you mentioned its ability to easily transform. Can you describe that?
Okay, so transformation is basically where we're putting in foreign DNA into the plant, and we do that using E. coli. We put the DNA into the E. coli, and then we extract that DNA. And the reason why we're putting it into E. coli is so that it can be replicated, so we can get a lot of DNA to put into another bacteria, which is called Agrobacterium. And the Agrobacterium is what puts the DNA into the plant. It puts it into the flowers so that the DNA can get passed on to the seeds. William?
Is the DNA, was it solely just so you can knock that gene over there?
Could you repeat that again?
Well, so like in part of it, you said that just in the DNA would knock out the gene. Yeah, sequence, but was there any other reason you did that? Like, I think I saw it, but I'm not sure if you said like application in PCR.
Okay. Well, I mean, that's sort of separate. What we're doing in transformation is we're engineering, well, not engineering. Well, we're creating artificial microRNA lines, which is something totally different. But what I was doing when I was isolating DNA was I was taking samples that we got from a database, which are mutant lines, which have tDNA insertions. When we're transforming, we're not adding tDNA insertions. brings about the phenotype that we see. So it's sort of general. Erica?
Why did you specifically use E. coli versus other bacteria?
Well, I mean, it's commonly used in transformations, but I mean, there's like a mini prep kit, and it's all ready for E. coli. So, Shalini?
How do you harvest the seeds?
Okay. So in Arabidopsis, what we do is we take the siliqua, which is the seed pods, and we rub them to release the seeds. In tomatoes, we slit open the tomato and then we scoop out the seeds. Nick?
How did the results differ for domesticated tomato and wild tomato?
For the root gland? Okay. So the domesticated tomato is much longer than the wild type tomato, and we can see that in the graph because in the graph the mean of the Pinelli root length was shortest, but M82 was nearer to the middle, so that's how they differ.
How did that affect the results?
Well, the thing is that the reason why we're measuring Pinelli is so it's sort of like a control because it is supposed to be statistically different, and so we're comparing ILs like 3, 4, and 4, 1, which were supposedly statistically different to how much statistically different Pinelli was. Does that make sense? Maura?
So you talked about the cortex levels in the beginning. Can you explain that a little?
Well, it's just cortex layers, which just means that Pinelli, it has 2 cortex layers. It's thinner.
Short?
It makes it thinner as opposed to shorter. And Lycopersicum has 3, so it's thicker.